name's James Sermani.
David Shore.
Ainsley Lockhart.
My name is Jerry Diltz.
My name is Irene Kaplow. I just finished my first year of my PhD in computer science at Stanford.
I'm a junior at Wesleyan University in Connecticut.
I just graduated from Georgia Tech in Atlanta.
Virginia Commonwealth University.
I am a junior at Kenyon College in Ohio.
So interns are important because they bring youth and energy to the JGI.
So I'm working with Dr. Grigoriev on fungal genomes, working on annotation of genomes based on repeat sequences and other things. So they actually have a pipeline that they use to run through all the genetic data, and so hopefully our annotation programs will eventually become a portion of that pipeline, and so we can just integrate it into the process that the JGI already has in place for fungal analysis.
My job is on creating a new function that measures average nucleotide identities between bacterial genomes.
I am working with an algae called Chlamydomonas, and I am basically making transcriptomes and seeing and stressing it out under different conditions and seeing what it's expressing.
I'm working in public relations, which I've now learned basically means defending what JGI does to the people who fund JGI. So looking to Congress, looking to the Department of Energy, and kind of trying to show that what we're doing here is in line with their mission.
So I'm doing a project on metagenomics, specifically metatranscriptomics. So we are now, in addition to sequencing many genomes, we can also sequence the RNA that's getting expressed in many of these organisms to figure out what kinds of proteins are being created in different environments that we know very little about.
I've gotten really good at Perl script. Really good. And I've learned a little bit of SQL.
So what I did this summer is I grew my algae without any manganese, which is an important component of several complex metabolic processes in photosynthesis. And so when you starve the cells of manganese, photosynthesis breaks down, and you can look at what enzymes that affected and what proteins that affected.
Well, I've actually learned a lot from my project. Our project has basically been on classifying repeat sequences in fungi, so I knew absolutely nothing about it beforehand. So I just, having, reading background papers, working on the code, talking to other people in our group, I've just learned a lot of what goes on in I guess, in fungal research, especially here at JGI, and that's been completely new to me, so that's good.
I got to write a press release about a paper that was published by some JGI collaborators and some from other labs. I basically got to read through this science paper, which is something I definitely would not have understood otherwise, and just kind of dissect it and figure out exactly what the key points were, and kind of trying to use that connection to people's regular daily lives.
So we have about 2 million genes in the cow rumen, and only about 1 of them have substantial sequence homology to known genes. So the goal of my project was to help figure out what the other 1 million genes are.
When I got the program working, karate chop in the office.
Sometimes the women I work with bring in their small children and I get to play with them. I'm serious, that's always the best.
Okay, so another thing that happened that was super funny that our intern did was we had it was like a outside event where we were selling ice cream and stuff like that, and our intern, he was the one handing out the ice creams, and I'm sure he heard them say, don't eat the ice cream right away because it's frozen and there's hot ice in the cooler, so don't eat it because it'll burn your tongue. He was the one handing them out. I swear he handed out about 100 ice cream bars. He was the one who opened the ice cream, and proceeded to eat the ice cream, and the ice cream got stuck on his tongue, and that was hilarious.
So that's what we're working on. More questions?